All right, so for those of you who aren't familiar with optical remote sensing, basically it allows us to examine landscape scale distributions in processes and wetlands. That can include things like vegetation type and health for mapping those, mapping land changes, mapping water quality, etc. And this is most typically done with multi-spectral satellite instruments like Landsat or MODIS. And how those instruments work is that they measure radiance and reflectance in several broader bands across the visible to shortwave infrared part of the electromagnetic spectrum. So this is a rendering of Landsat 8, for example. And then this figure up in the right shows the spectral signatures of a few different land surface types. So water, you see, basically just reflects in the visible portion. You see blue, green, red highlighted. Soil has a pretty even reflectance spectrum across for the visible, increasing through the sphere. And then this is a very prototypical looking vegetation spectrum. So I'll come back to this a few times throughout these slides just to reference. But you can see in these gray bands, these are the very rough approximations of where the Landsat 5 thematic mapper bands lie in relation to the spectrum. So you can see there's a lot of, well, plants absorb most of the visible part of the spectrum, so the reflectance is lower. But even so, the green, they have higher green reflectance than blue and red. So you can see it absorbs more in this little trough. And then when you go into the near infrared, the chlorophyll content reflects a lot of that incident radiation. So they reflect a huge portion of it. And then onwards into the shortwave infrared, that's often related to things like moisture content within the canopy. So that's optical remote sensing and multi-spectral instruments very generally. But what then is imaging spectroscopy or hyperspectral data? And the difference is, instead of measuring across those discrete, broader chunks of the spectrum, we're making continuous radiance measurements from which we can derive surface reflectance. So when AVIRIS-NG flies over a surface, in each pixel, it's measuring across over 400 bands to reconstitute sort of the reflectance properties of that pixel. So this is a section of an AVIRIS-NG image sample that we can call an image cube with that stack of bands visualized behind it. And within each pixel there, we have a spectrum like this from the surface reflectance product. And so estimating the full spectral reflectance of a surface is really useful because you can think of it as a actual property of the surface rather than just a simple measurement. And that's very closely associated in different parts of the spectrum with different plant structural and biochemical properties. And those unique spectral signatures also enable land cover and vegetation mapping with greater detail than our typical multi-spectral sensors let us. And other studies that you can apply this to are applicable to the atmosphere and also water quality. And so with an eye to the future, also AVIRIS-NG is a good stand-in for the upcoming surface biology and geology mission. And a lot of the science that's been done with AVIRIS-NG directly informs the design of that satellite instrument that's right now in the planning phase. So this is an example of one of the groups of flight lines that we collected for the spring campaign part of Delta-X over the Atchafalaya Basin. So these are all the vegetation flight lines that were collected in succession to capture the Atchafalaya and Wax Lake outlet complex. So in general, the AVIRIS-NG data that we collected, it spans from about 380 to 2,500 nanometers. And that's 425 bands in these data sets at a 5 nanometer sampling. So we also flew the airplane at about 5,000 meters to give us a roughly 5 meter spatial resolution. So like was said before, there's 3 airborne campaigns conducted last year. So late March into April, August, and then September after Hurricane Ida hit. So this, obviously the wetlands here are very spatially complex. You can imagine there's narrow channels of water surfaces that cut through the wetlands. There's a lot of mud flats that are exposed. And so these all these sort of ecological factors have a big impact on the spectra that you measure over a particular pixel. So it's a very spatially and spectrally complex region. So AVIRIS-NG really gives us a ideal data set to try to disentangle that information and make more accurate maps of vegetation and their properties and water quality, because we can better control for the sort of muddying impacts of all those wetland complicating factors. And so this is the processing pipeline generally for the AVIRIS-NG data. So the L1 product that it measures is the radiance at sensor. And then we apply an atmospheric correction to transform that data, remove the atmospheric effects, and return the surface reflectance. There's also additional corrections that we do to remove impacts from the bidirectional reflectance distribution function and water glint over, or sun glint over water pixels. So it's a little hard to see in this image because the data is actually pretty clean to begin with. But the BRDF essentially can cause variations in the illumination intensity across track within a flight line. So if this is all dependent on the sun angle and the instrument relative to the instrument viewing geometry. So if you have a lot of BRDF effects, one side of the image will look much more shaded than the other side. And so when you start stitching the images together, it'll give you a lot of discontinuities that need to be corrected, or your mapping results start to return errors and it looks very disjointed. So the BRDF and then the sun glint, that's the BRDF, and then also the sun glint happens when especially when the sun is too high up, it reflects more directly off the surface and strikes the sensor like a mirror. So correcting some of those effects also allows us to better map a lot of the water quality properties. So yes, we applied those corrections, and those data are L2B, and those are also all be available, and all the subsequent L3 vegetation and water derived maps will be based on those. However, for the lessons here, we're just going to use the L2 surface reflectance data because like I said before, the data is already pretty clean. The flight line we use is in good shape, and it was available in preprint form prior to the L2B data. So you don't really need to worry about this, but if you're going to carry forward and use the AVIRIS-NG data in your own studies, I would recommend using the corrected L2B data. So this is an overview of how much data we collected, which is a lot. It's hundreds and hundreds of gigabytes. So across the spring, fall, and post-IDA deployments, we have 144 terrestrial vegetation flight lines. So this is our full coverage of those flight lines. In the spring, we collected all of these boxes once over. In the fall, just the Atchafly and Terrebonne boxes here, because I'm sure most of you know what August in Louisiana is like, and there's a lot of cloud cover that sort of impeded the flight operations. But we made do, and they're actually pretty good products in the end. And then in post-IDA, when we came back in September, we also collected all of these boxes again. And in addition to those lines, we collected 44 water quality lines that cut across the Atchafly and Terrebonne basins. So again, these are all the products available. I'll also mention, in addition to the individually corrected flight lines, we have L2B, glint, BRDF, and glint corrected mosaics. So this example that I showed earlier in the Atchafly and also the Terrebonne here, those will also be available, and those are already stitched together and ready to go. However, they are very memory intensive. This one here is about 200 gigabytes, and this one here is about 400 gigabytes. So it requires some creative programming to handle but also they're not very GIS friendly. But if you have access to programs like Ndvi, those Ndvi can handle it pretty well as well. So I'll get on to the first one or two modules of the workshop here. So the first one, we're just going to just get a feel for the data essentially. Open the data, visualize and export bands, plot some spectra to basically dig into it a little bit. And then we're going to do basic applications with NDVI, and then a little more complicated operations with principal components analysis that makes use of all of the band data that we have, and then feed that into some basic classification operations. And lastly, developing water quality models to map total suspended solids with a partial least squares regression approach. So yes, this is the first one, just the data introduction. So with that, I'll switch over to my Python notebook. And I have to apologize ahead of time here. I'm going to, unlike Alex's presentations, I'm going to drive from my Jupyter notebook here because in the part 3, there's a memory-intensive operation that doesn't really agree with Google Colab's RAM limitations. So you can follow along in Google Colab. If you're feeling adventurous, you can also just, and you have all the data installed locally, you can follow along in Jupyter Notebook. But feel free to just stick with Google, and when we get to that, we'll cross that bridge when we come to it. And you can follow along with the lesson then, and then you can reopen the actual finished data product that I have uploaded on the Google Drive after that part of the module. So you won't have to, crash Google Colab yourself. Okay, so does everyone have the Google Colab or Jupyter Notebook open for this module? Okay, so to start with, all of these modules are just based on one flight line. So that's hosted in the Google Drive, or you should have downloaded it ahead of time yourself along with the corresponding header.hdr file. That includes important metadata information for that flight line. Again, this is an L2 product, and this is the one we're using. The date nomenclature for these files, it's ANG, and then it's year, month, date, and then timestamp. So that's right in the file name. It tells you when exactly it was collected. So we'll start with just importing the libraries. For this one, we'll use GDAL, OS, NumPy, and matplotlib, and then later we'll also use sklearn. I want to note here, when it comes to GDAL, it might depend on your specific installation, how GDAL was installed, but you might need to change this to from OSGeo import GDAL. So if you get an error thrown at you in your imports, that might be why. So just try changing that line. And then this block here, this is the same thing we've been doing for the previous modules. I have it commented out in my own Jupyter notebook, but this should be the default that you run all ready to go in the Colab version. But I'll just run my local version here. So this basically just, this is my file directory where I have things saved, and I designate an input and output folder, and then designate the file path of the actual data file that I'm opening. And it's the equivalent of everything in the Colab version. Okay, so the first thing we're going to do is just open the file and just pluck one band out of it and visualize it to just see what it looks like. So I have it designated as pulling out band 57. So I just chose that one, and it's a red wavelength that corresponds to the center of the Landsat 8 red band for reference. And you'll notice that the data structure here, the flight line is saved straight up and down, when in reality, it's actually sort of diagonally oriented. So that information is contained in the header file. So some GIS programs will be able to read it and plop it down in the correct location. But I have noticed that not every utility or program wants to read it in and put it in the correct location. So sometimes it takes a little extra. So yes, these values are unitless actually. So this is their reflectance, unitless reflectance. Oh, I'm sorry, on the axis labels. So those are just like pixel numbers, essentially. So does everyone have the band loaded and visualized here? So I have it just plotted from, so we set a min of 0 and a max of 0.2. But there's other things at play here. The NAND values, which we went over in the tabular data previously, they're all saved as -9999s within the image data. So Python will read this image in, and the 999s are still present. So I just set the minimum to 0, and it'll ignore those. And the max is 0.2. If you remember from the that sample reflectance plot in the PowerPoint, the visible wavelengths reflectances are usually on the order of 0 to very low, 0.1, 0.2. So setting a max value there helps visualize it more. If you just let it default, you're probably just going to get sort of a very washed out looking image, and you won't make out much detail. So these pixels are about 4.8 meters. Yep. Yes. Mm-hmm.
So what's the second question? Just asking what's the spatial resolution of this map? Just trying to compare it to Landsat 8.
Okay, so that's just a single band. So it's useful information, but obviously we want to look at what all the data at our disposal looks like. So if we run the next cell, what I have here is just plucking out a single pixel, and we're plotting the spectrum from that pixel. So I picked just these coordinates, 500, 5400. So you can see 500 here, 5400 here. So I chose it because it's in this sort of forest patch in this island in the Atchafalaya Delta. So it would give us just a good representative vegetation spectrum. So then these are just lines for making this plot figure. So you should have this pop up. So you'll notice a lot of those things that we went over before in the introduction kind of pop out in this figure as well. So you see the visible section here, higher green reflectance, whereas more blue and red were absorbed. This is the red edge, and then the near infrared, and then the shortwave infrared regions. You'll also notice, obviously, there's these big spikes of noise present between the near infrared and the shortwave infrared regions. Basically, these are atmospheric windows where water vapor in the atmosphere is absorbing almost all of the incident radiation at those wavelengths. So the instrument is still trying to read reflected photons from the surface, but those photons at those wavelengths are actually all being absorbed by water vapor in the atmosphere. So what comes out is essentially noise. So to be able to make proper use of all the data here, we construct a bad bands list. So basically that's just a binary list, ones and zeros, that tells you which bands are good data, which bands are bad data. It's rather self-explanatory. Also, this line I just have designated the full list of the wavelengths in nanometers for each band that we have read in. So these are the, so I nix out some of the tail ends of the data where there's noise, but primarily these two chunks, these indices, index ranges here, nix out these atmospheric windows so that we get rid of a lot of this noise. So then if we plot the next one, we plot the same pixel, but we're setting the all the values where the bad bands list is set to 0 as nans, which we also went over previously. In addition to that other one, I'm also plotting a water pixel just from coordinates 200, 200. So plotting those together, you can see what these different surface spectra look like. So this is the that same vegetation pixel with that noise removed, and then this is the water reflectance pixel. This is also a pretty prototypical example of what a rather turbid water surface looks like. And then the last thing here for this module is we just want to save some of the data as to construct a true color composite. So we're just going to take out the red, green, and blue bands. Before we do that, I think everyone should probably open QGIS and let it open so that we can then load it and visualize. Yes, there's a question?
Yeah, there's a question online. Did the water pixel have to have the same bands removed?
So, yes, but one thing you'll notice with the water pixel is there's really only useful information through about 1100, 1200 nanometers. Beyond that, pretty much any water surface absorbs all of the incident radiation. So there's virtually zero reflectance past that near infrared. So yeah, you'll notice we should remove those bad bands if we want it just for a consistent comparison. But if we're just doing water, if we're only looking at the spectral properties of water, we're going to typically remove everything past about 1100 nanometers anyway. Another question? So this
Can
one.
you repeat the question, please? What type of atmospheric correction is used for this?
So this one is an atmospheric correction algorithm developed by David Thompson, who's the guru behind all the processing of the AVIRIS data. And it's this not my specific area of expertise, but it's an optimal estimation algorithm that ingests a lot of the field in situ field data to sort of match up the actual measured surface reflectance spectra with what is measured by the aircraft instrument. All right, so does everyone have QGIS open? Okay, so we'll run the last block here. So what this does is it just grabs bands 57, 38, 22. And again, those just correspond to the Landsat centers for red, green, and blue, respectively. And I have them printing out just the wavelengths of those bands. So I referenced that wavelengths list and then the proper index number. So this is the wavelength in nanometers that those bands correspond to. And then this block grabs all the relevant geo information from the input data that we opened. as well as setting the output information, and writes it as a new data set. So if you're in Collab, it should write that out to whatever you have your output folder set to. So hopefully that appeared in there. And you might have to download that to actually open it in QGIS then. For my own one here, I have an output folder that it writes to. So I just have it saved as rgb.tif here. And then if we open it in QGIS, drag it in, probably won't see it look that neat right away. Question?
Are they properly saving in the Google Drive? Has anyone? No, they're not.
Okay. Okay, so sorry, I should have covered this when we were doing the imports, but if you're doing this in Collab, as I mentioned, almost everyone is, you should set this line here where it sets the output directory to whatever folder within your own particular Google Drive you want to save your data to. So right now it might be trying to save to the shared folder, which is why it's not outputting correctly.
Yeah, so you won't have saving rights to the shared drive, so you should change that path to, it should basically be /content/drive/my drive. That part can stay. And then, in fact, maybe just use that and it'll just save to their drive.
Yeah.
So do you maybe want to change that so they can see the example?
So you could just do something like, yeah, so you can use just something like this or whatever, whichever your desired folder is within your own drive folder.
But they'll probably need to make that folder.
Yeah.
So if you remove output, it'll just save it straight on your main drive. Is that easier? Can someone try that? That's a very good question. Can you generate directories in your Google Drive from Collab?
I'm not sure. Yeah.
I will try it and let you know. Okay, has anybody changed the output directory and gotten it to work? Did you just use my drive? Okay, so yeah, like Daniel is showing, change the output directory to what it says right there, and it will save on your main Google Drive. Anyone have issues with that?
To which part? Oh, sorry. This is the import, or the one that sets up the Google Collab. So it should be the second cell block.
Okay, so I tried to make a directory and it seemed to work.
Okay.
If someone wants to know how, you can ask me afterwards. It's pretty simple, but for now, just save it on your drive. Yeah, so it should be this, the second cell. It's where you're authorizing the Google Drive. There's a line that says output dir equals, and you want to match exactly what Daniel has shown up there.
Yeah. So if you have that set, try rerunning that block, and then assuming that goes correctly, just rerun the one at the end, and then see if it outputs rgb.tiff in that folder that you designated. And if it did, then I would download that TIFF file. It should just be something like 80 megabytes.
Daniel? Daniel,
Back up.
yeah. When you answer a question, can you please just repeat the question first?
Oh, yes.
Yeah, if you're having any issues with this, the cell where you are outputting, you need to go back to the second cell where you authenticate Google and change the output directory to content/drive/my space drive. You need to rerun that cell before the others will work. Hey, Daniel, what is the file that will be output so they can check if they...
It's rgb.tiff. Got it? Cool.
Sorry. Are we going to open it in QGIS then?
Yes, yeah. So once you have that output into your Google folder and then downloaded onto your desktop, drag it into QGIS.
Right. And it looks something black and white to me.
Yeah, yeah. I'll get to that once everyone is on the same page. Okay, if you're getting errors at data set out, then it probably is trying to, it probably has an improper file path that it's referencing.
Changed to my drive and successfully got that first script to run.
Okay.
Do I have to run every script after that?
You should just be able to rerun this one, but just make sure they run all properly. First, yeah,
go
try it
back
from the
and
top.
run. Did the first one, it ran after I changed it to
Okay,
my drive.
yeah,
Didn't have
that
to,
looks right.
but I didn't. I jumped forward, so I'm just going to run them all.
Yeah, it doesn't hurt.
Maybe it'll. That's going to solve it. It liked to. That's what it was. Had to run them all.
Cool. Okay, so it looks like most people are done. Can I just get a show of hands who still is having issues with outputting the data?
There's a question about if you do run all, it will crash. And that is because there are, I think there's 1 or 2 cells that are memory intensive.
Yeah,
and
it's
won't work
gonna
on Colab.
run you through all of the modules then, and it'll take you through to the end. But Google Colab won't take too kindly to 1 or 2 of those in the later modules. Okay, so does everyone have the file downloaded and open in QGIS here? Okay, so if so, you probably just see an image that looks like black or black and white or something like that. And that's again probably because of those NaN values here. So to just visualize this in true color, just go to properties. And then it should be align red band, band 3, green 2, blue 1. And change the min and max to 0 and 0.2 for everything. So that's just setting the color scale for each of those bands, 0, 0.2. So it'll disregard the NaN values and any errantly high reflectance values so that we get a better stretch to visualize with. And then it should look like this. And it should line up pretty well with the base map if you have it loaded. Yeah. So right click on the file, properties, make sure multi-band color is selected here in the render type. And then make sure red, green, and blue is band 3, 2, 1 respectively. And then stretch it from 0, 0.2. You can play around with the stretch if you like. You could also reorder the bands, test it out if you want to see what different false color composite combinations look like. But I find it helpful and we'll reference it in the future to make a true color representation with a nice visible stretch on it.
The question was to go over the last step.
All right, it looks like we're ready to pick back up. So before I pick back up, did everyone get the image properly exported and brought into QGIS? So you got something like looks like this, or perhaps just with a black bounding box all around it, same difference. All right, so I think all those problems are sorted out, so we'll carry on. So next step, we're gonna just do one of the sort of staple optical remote sensing product and analyses, which is just make a normalized difference vegetation index. So here we're gonna calculate an NDVI image. You can see an example of this from a previous data set here. Plot a histogram of the NDVI values and use that to generate a land-water mask for that flight line that we've been using. So the normalized difference vegetation index, it's essentially just this very simple normalized ratio between the near infrared reflectance and red reflectance. So if we again look at this curve with special attention to the green vegetation reflectance curve, we have our near infrared band up here that has a higher reflectance value, and the red band that is usually positioned in the bottom of this trough that has a low reflectance value. So again, chlorophyll typically reflects a lot of the incident radiation up here so that the plant doesn't overheat, lose all its water, and die. But red is photosynthetically active radiation that it absorbs almost all of. So you can leverage the difference between those two with this ratio to come up with a proxy for plant health or primary productivity. So the applications of NDVI, they include monitoring vegetation phenology, so how plant activity changes over the season, assessing growing season length, general plant health, etc. So this, again, this part of the spectrum, this is called the red edge. That's the transition zone between where the plant absorbs the visible radiation and reflects a lot of the near-infrared radiation. So that's a sharp increase at about 690 to 730 nanometers. Yes, question? Oh, yes, I'm sorry. This one? Is that better? Yep. So that sharp increase in the high reflectance in the near-infrared is a measure of essentially the greenness of a pixel. So the NDVI range, when you calculate that ratio, it generally goes from -1 to 1, where a brighter pixel with values closer to 1 are more green with more photosynthesizing or healthy vegetation present there. So you can see just sort of a example of how this calculation works in this picture. So in a healthy tree here, you see it absorbs a lot of the incident red radiation, reflecting only about 8%, but reflecting 50% of the near-infrared. So that gives you about NDVI of 0.72. If it's senescent or the colors have turned to fall and the chlorophyll is kind of died off as the leaves are going to get ready to fall off, it reflects a lot more of the visible because that chlorophyll isn't there anymore to reflect that photosynthetically active vegetation or radiation. So it's reflecting 30% of the visible then, and also reflecting less of the near-infrared. So the NDVI here is 0.14. So it's a big market difference. So that's just how NDVI works. So we're going to go back to the notebook or your collab and just make one of these images ourselves. So everyone's collab is still like where we left off. So everything is still read in. We don't have to rerun anything at this point. So the first thing, we're just going to open as arrays the red and near-infrared bands. So that's bands 57 and 99 here. So we'll run that. And then also I'll use the boundary of the image to generate a mask. So I just use the red one here and take the values where we have it marked as the NAND value of -999. And I just use that to make this binary mask where 1 is here, 0 is on the boundaries, so that we can only just, we can reference that in the future to just use the values where we actually have those reflectance units. So that's what this is here. Oh, it should be showing my screen. Let me try again. Oh, okay. Let me expand this too. So then we just use the near-infrared and red bands that we have read in and just calculate that simple ratio here. So near-infrared minus red over near-infrared plus red. And because the -9999s kind of mess with those boundaries a bit, they'll come out as different numbers. So I just enforce that where the mask is 0, just enforce it as the NAND value here. And then we use the matplotlib and set the scale from -1 to 1. And I like this color map. It just represents the NDVI values a little better and show the values here to come up with this map. Does everyone have this plot or something equivalent? Okay, so you can see very clearly water has very low NDVI, plants very much higher NDVI. And then this one here, we're just going to save the NDVI file so that you can also open it in QGIS. Same as our previous imagery. So if you drag it in the same way, Well, kind of misplaced mine. There it goes. It should show up by default as sort of a grayscale image, but you can change the stretch values here. So why don't we change it to single band pseudo color? We'll set it -1 to 1, not 21. And pick your favored color stretch here. I'll just choose something that diverges in the middle, like this one. So you can reference that in the future when you trying to interpret the image or overlay other classes for your future classification on it, that kind of thing. All right, is everyone there? Cool. So the next part here is we're going to use the actual NDVI values to make a land-water mask. So this is really simple. First thing we want to do is investigate the distribution of NDVI values in the image. So this line here, we're just extracting the NDVI values where the mask equals 1. So again, just excluding those NAND values in the boundary and plotting a histogram. So I used 100 bins to give it a little more detail. So this standard histogram distribution, the y-axis is the count of pixels within the bin, and then the x-axis is NDVI value. So what we're looking for here is just sort of a value to cut it off, below which it's almost certainly water, above which it's almost certainly land. So it's usually around 0, because if you have land with where the near infrared reflectance is higher than visible, it'll give you a positive. Water reflects more visible radiation, but absorbs almost all of the near infrared, so it's almost always negative, unless it's extremely turbid or you have submerged aquatic vegetation or something like that. But don't worry about those sort of complications for now. So that value close to 0, where it sort of diverges, I've honed in around 0.05 here, this little break. So we can work under the assumption that everything to the right of there is land, everything to the left is water. So that's why in the next block here, we just do a very simple 2-class density slice. This is sort of density slices, basically the most basic form of classification you can do. It's basically just using threshold values for band reflectance or an index like NDVI, and labeling those thresholds as different class types, i.e. land, water. So I have selected 0.05 here, so we're just going to create a new array, just empty with zeros in the same shape as the original one. And everything above 0.05 NDVI is going to be 1. And then again, use those mask values to enforce the NANDs. So if you plot that, you should get this spit out. And then we can run the last one, same as everything else, to export the TIFF image. And then we can bring that into QGIS and you can inspect. So you might not, there might be some areas here that might be a little iffy because a lot of these deltaic islands have like their tidal mudflats. They might be inundated that much, and the instrument might not be able to discern that much water with the mudflat underneath compared to an exposed soil flat. But overall, it does a pretty good job here of just discerning land from water. So you can look that over briefly here. That's true.
Do you have them saved somewhere so we can do it fast or no?
Yeah, I can pause here to catch up.
Yeah, because we have to like.
Yes. Sorry, I should pause when we have to drag these things into QGIS more.
All right, is
So let me
anyone
download and
having
catch up.
issues? take a minute to go around.
Someone online asked he's not getting the colors to show properly. Do they have to edit the symbology?
Not getting the colors in the NDVI file.
It's not specified. Oh, yes.
In the QGIS? Yes. So if you right click on it and go to properties, just like we did with the RGB file, and you select single band pseudo color under render type, you can pick a color scale and scale it from -1 to 1. If you prefer, you can also just make it grayscale with single band gray the same way. But usually with this, I prefer to use a diverging color scheme like this.
Hey, Daniel.
Yep.
How did you get that beautiful map behind it?
Oh, so that's a base map.
I think most people don't have that. So if you could just explain.
Yeah, so I don't know if QGIS comes pre-installed with a plugin for a base map, but the one I use here is HCM GIS. So if you go to your plugins and manage and install plugins.
I think you can get at least something under the browser.
Okay. So this is one benefit of QGIS is there's a lot of open source plugins for all sorts of utilities, including using base maps. So that's where I got that one from. Sorry, Talit, was that a question? Oh, yeah. So it's plugins, manage and install plugins. And then I believe this one is HCM GIS. So you can select this one and just click install here. It'll be automatic, and then it'll give you another pull down menu here that you can select a base map from. So the one I use here is just the standard Google satellite imagery. Yep.
Or that it doesn't save automatically. So if you made a change and then you close a tab and reopen it, it won't still be there. So if you make edits and you want to save it, you have to save it to your own Google Drive.
Yep.
Use 0.5 for NDVI to identify water and land. Could you introduce a little bit more? How do you find this value?
Oh, yeah. So I'll go back to the histogram here. So in general, the dividing line should be very close to 0, if not 0 itself. And I just chose 0.05 because that's where this inflection point in the histogram is. You could just use 0 as a starting point and inspect it. It should only be very slightly different.
Okay. Do you think the very turbid water will influence this threshold?
I'm sorry, can you say that again?
Do you think the very turbid water, which has more reflectance for near infrared?
It can. The main part where it might confuse it is you might confuse very turbid water with exposed mudflat, one being technically inundated and one not. So there might be some confusion there. But in general, the water should still absorb more near infrared radiation than an actual exposed mudflat should be. So they should still have some different spectral signature on average. No problem. So just for symbolizing the NDVI. Yeah, so it probably didn't come up with a very neat looking image when you dragged it in, right? So you just want to right click on it, go properties here, and I choose under render type, single band pseudo color. And then I set the min to -1 and 1 max to 1, because that's the theoretical limits of the NDVI values. And then here under color ramp, you could just pick whichever one you like. That sort of fits the what the symbology should mean. So high NDVI values mean like healthy vegetation, and vice versa with negative. So I generally choose something that reflects that.
Hey, Daniel, can you go over the mask again, the coloring?
Yeah, sure. The coloring for the mask.
Yes,
This?
please. Yes.
So I didn't change this one, I don't think. It's just set black to white, 0 to 1. Yeah, so the one I used, I think Mark also posted a suggestion for the plugin he likes in QGIS. But the one I use here is HCMGIS, and you can get that by going to plugins, manage install plugins, and searching for that here. Then you just click the install button and it takes care of everything for you. Might be a versioning issue. You can just search base map in here and you could probably find several different options as well. Oh, it didn't give you this pull down menu. So once you install the plugin, it should give you this pull down menu. I hope you don't have to restart it or anything to pull it up. And then this is the one I usually use, just the Google satellite. The base map is very helpful to just as a visual reference, but it's not strictly necessary for doing any of these module exercises. But it is helpful to make sure that the imagery is overlaid in the correct place and all the georeferencing is right.
Transparency within properties. There's a little select tool, little mouse, and if you click where it's not, you've already done it, but yeah, then it'll tell you what that value is.
Again, if you can't get it to go away, I wouldn't worry about it too much. The more important thing is what's in the data frame itself. Question? Oh, yes. That's, yeah. So the question was to, again, how to get rid of the bounding box if it shows up and obscures the base map underneath. So we can go to properties and transparency and find what the no data value is, because that's what that's what's showing up in the bounding box.
So in this case, it's probably both -9999 and also 0. So you'll have to enter both of them.
So the important thing for this module to just get done and saved is just make sure that the NDVI mask file here is saved in your folder and output correctly, because that'll be referenced again in the future modules for applying a model. Oh, do you have it done? Yes, exactly. If you pull it up, you're good. So I'll move on to the next module, which is principal components analysis. So this is the one that'll have memory issues on Google Colab. So you don't have to run these commands if you're operating Google Colab. If you're on a better computer and you're running on Jupyter, you can feel free to follow along with the commands. But otherwise, I'd recommend just just follow along here. And then at the end, I have the PCA output saved in the shared folder. So you'll just download that one directly. So the objectives here are to perform a PCA dimension reduction on the image, plot the explained variance from the principal component analysis, and visualize the component bands and export those output selection, like this example image here. So PCA is a really common linear dimensionality reduction technique, and it's really well suited for high dimensionality imaging spectrometer data because we have 425 bands in each image that we're working with. And a lot of those bands, especially as they're more adjacent to each other, are collinear with each other. So you can't just use all those bands and split them into regression algorithms or classifications without getting redundant information and skewing your results. So this figure in the upper right, this is a covariance matrix. So this is a visualization of how correlated each band is with every other band. So brighter white is a stronger positive correlation, and darker black is a stronger negative correlation. So you can see here, this is like this first square, the visible bands are very correlated with each other. Then you move into the near infrared and they're less correlated with each other, and so on and so forth. So a principal component from the transformation is a projection of that original data that captures the variance inherent from the original data set. So the first component always captures the most variance, and then components are uncorrelated with each successive component that you add onto your principal component analysis, such that correlated features in the original data contribute to the same original principal component. So every principal component from the output, each successive one should be orthogonal to each other in the principal component space. So they're all non-collinear. They don't have any overlap at all. So you can treat them all as independent variables. So PCA for imaging spectrometer data like AVIRIS-NG is really useful because it can reduce the correlated bands while explaining the vast majority of the variance in the image. So you don't have to use all 425 bands in whatever you want to do. And it's useful for capturing and describing different surface properties and enabling efficient improved classification, as well as potentially other algorithms if you want to feed it into a regression model or something like that. So this is an example from another paper we did where we did a PCA to feed into a classification scheme. So these are the principal components visualized. So you can see what you don't need to look too great into detail, but you can see that there's a lot of different sort of biophysical effects that the principal component transformation is picking up on here. So you see bright white values here over the water here. So it's almost certainly picking up on like water content, not just open surface water, but probably like canopy water content, stuff like that. This is probably picking up on, you see bright pixels in these tidal flats, so it's probably picking up on something in the soil. So I'll switch over back to the Python notebook here. So I already set these first two cell blocks to run. But basically what this first one does is it references the mask in the bad bands list. And it's using those to construct a new array and feeding all the relevant values in. So I tried to make this as I tried to reduce the memory intensiveness of this operation as much as possible so that I'm not reading in excess information. So what I did is it's cycling through all the bands that are designated as good from the bad bands list. And then it's also referencing the mask. So it reads all those values in where the mask is good. But because the AVIRIS-NG data is floating point data, that's more memory intensive than just integer data. So I multiplied the values by 100,000 so that I could save them as integers. So when we do that, it's less memory, but the output statistics will be the same. And then this one actually applies the Pca transformation with the sklearn module, or the sklearn library. So it just transforms the, it puts the axes right to feed into the Pca function. And here we're going to project 10 components. The default number is usually how many bands or features are in your data set. So here I have 300 designated good bands, but that's totally excessive. So we just have 10. You'll almost never get to like 10 principal components that actually have usable data in them. So it fits the Pca transformation to those image values and applies to the transformation then. So then we'll run the next one here. So this first block here is we have the image transformation done here, and we're just mapping it back into a sort of map-like array. Sorry, was there a question?
Out of the 300 bands you have chosen 10 bands, of which 10 bands are?
Oh, no. So we're doing a transformation on those 300 bands, and it's transforming them into 10 new like synthetic components that will explain all the variance inherent in those 300 bands. Does that make more sense?
Okay, thank you.
Yeah. So we're creating a new synthetic data set, but like condensing all that data into a more manageable size that explains the same sort of biophysical properties that the original data does. Yeah, so here we're just mapping the output components back into sort of a map shape. And then here we're plotting the cumulative explained variance by component. So like I said in the slides, the first component always explains the most variance in your analysis. So it's almost 99% here. I have it also listed out here. So 94.3%. The most common selection of components is to take the selection such that you're explaining 99.9% of the variance. So that corresponds to the first 6 components here. But you can see, like, as we go along, we're basically just approaching an asymptote in terms of how much variance we're explaining. So you don't want to select everything, just how much is useful. But beyond just how much variance you're explaining, it's also helpful to visualize your components. So here I have a plot of all those 10 output components. So you can kind of see what information it picks up on. One thing you'll notice is that the further along you go, just sort of the more gray and washed out it gets, which is sort of what you would expect given this plot, right? That you're not explaining that much more variance. Yes, another question?
So you're running this PCA on just a subset of the bands. So you had that like bad bands filter. Yeah. Is that just something that you know beforehand, or how did you define that?
So,
and what would it have looked like after the PCA if you hadn't removed that stuff?
So if we didn't nix out the bad bands, we're basically just introducing more noise into it. So that was one thing I wanted to get at with this very first plot that we made here. There's a lot of bands within the stack of the original 425 that are just noise because the atmosphere is absorbing all the useful information, right? So we could keep all of that in, but it's not going to add any useful information to our actual transformation. So the output should really look the same, but it's just going to take more memory and possibly, skew the results a little bit. So yeah, we just want to narrow it down to just what is useful, relevant information as much as possible. But it is kind of hard to determine that ahead of time without just sort of testing and playing around with the data yourself. Sorry, was there another question here?
Do you know after the reduction of bands, which bands are there in the PCA?
I'm sorry, can you say that again?
So after the PCA, how do you know like which bands are eliminated from the?
So how do I know which bands to use after we've applied the PCA? Yeah, so do you mean of our output component? Yeah, so the way I do it here is to select the ones that explain 99.9% of the variance. So that's the first 6. But I also kind of cross-reference that visually by plotting those output PCA bands here. So you can see this first set here pick up on some identifiable information. I mean, you can see the bright values here, for example. Maybe the 6th one isn't as useful, but we're still including it. But as you go further along, it just gets more gray and washed out. So there's not that as much useful information inherent in those other ones. So we can just leave those out.
I might be getting, oh, sorry, you have a follow-up? Okay. I might be getting into the weeds of just the PCA, but
yeah.
Is there a way to see which bands are incorporated into each PCA band without, so you could like go back and see what wavelengths might be like in PCA 1?
That is a very good question. There should be a within the PCA function itself, I believe you can output like an array of the loading values. I didn't want to get too far into the weeds for this, but yeah, that's a very good question. So to do that, yeah, you would output the loading values for each band. That'd be definitely relevant information.
Does anyone else have any questions for Daniel?
Okay. All good questions, so thank you. So the last one here is we decided to take the first 6, again, to explain 99.9% of the variance, and we output it the same way we did with everything else. And so this one should be saved in the output folder here. So if you want to access that and download it, then we can just bring it into QGIS, same way we've been doing with other data. So once you get this loaded in, I realize you probably need to wait for it to download and drag it in and everything. But this is just the default multi-band color symbolization that it brings it in with. But I find it really useful to just sort of interpret what the different PCA components are picking up on. So you can see the red band represents the first component, and you can see, obviously, it's very bright red in these healthy green vegetation areas, right? So that should tell you something about what information is captured by that component. And then you can just symbolize in different ways to play around with the data. Sorry, single band gray to explore the different bands here and see what they look like on their own. Or you could go back to multi-band color and just see what the different sort of false color composites look like to explore it further. But I'll just leave it as the default that it loads as with bands 1, 2, 3.
Hey, Daniel, can you remind us where this file is?
Yes. So the file should be wherever you set your, wherever you decide to save your output data at the top of module 1 when we activated the Google Colab. So I think it was,
Has
I
anybody
changed it
found
up
those
here.
files? Yeah, Daniel, so is this something they can generate, or is this the memory issue?
This is where you're going to hit the memory issue in Google Colab. So that's why I uploaded this one separately. But we're going to want the data output here for the classification module next.
So yeah, where do they find the files that you uploaded, and then where should they put them?
Oh, yes. So in that shared folder, so Delta Workshop 22, tutorials, AVIRIS-NG, data, and you should see these files in the output folder here. So download the PCA one here.
Yeah, so you should have been able to produce the RGB, NDVI, and NDVI mask on your own. Sorry, scroll up in that, double click.
Question?
From a workflow standpoint, like what's the real time consuming piece? Like I'm still chugging through just like reading in all of the bands on my laptop versus doing the actual PCA once it's in there. What's the
time consume? Well, those are the two most time consuming and memory intensive steps. So this one takes the longest, I think, from at least on my computer. So it's got to loop through all of the bands and read the data in. So this is a good computer. It takes like 30 seconds to a minute, but it could kill other computers in the process.
I think we did it in 3 on my not as great computer.
Yes. But then actually fitting the PCA to those data once you have it read in, it could also take a minute. But we tested it on the other laptop and that caused a memory error on that one. So yeah, just something you have to be aware of. This is one of the distinct drawbacks of hyperspectral data, and there's not really many good ways around it. You just have to sort of get creative with how you sort of dance around memory issues with it. Yep.
Are we just not seeing the variation that one's picking up? Or is it that
This
that
one?
small piece that's so important? Yeah. Third one, right? So
Yeah, I'm not entirely sure. It does look like it's picking up on some soil or turbidity a little bit, but definitely not as much as the first two or 4 and 5, for example.
Any other questions? Can everyone find the PCA file? Download it?
when you do your own analysis and depending on the application of the PCA, I mean, you could be more selective, more judicious about what actual components you select and carry forward based on what you want out of it. But just for this, it's just the most basic to just set the threshold and select everything that contributes to it. But I've done the same process where, like if we go back to this one, it wasn't just all in sequential, all of the sequential components. There was one or two in there that after visual inspection, like it looked like it was picking up on excess noise and wasn't going to be useful for the classification, so I threw them out. Yeah.
Is anybody else not able to find the file?
It's unitless. If you look at the values associated with each one, you can see that they're kind of scaled differently.
Okay, actually, I'm just repeating for the people, the internet. But my question was, what was the unit of the gray, like in those gray rasters? And apparently they're unitless.
Yeah,
but it was just like,
unitless.
what's the physical meaning behind it? So if something was light, like obviously that's good or interesting, but like,
Yeah, that means that it's a high loading value for whatever particular sort of biophysical effect that component is detecting, right? So if it's something like a plant health or a chlorophyll content, you'll see bright pixels over heavily vegetated healthy areas. If it's a water content within the canopy, same thing.
so with that, like a higher loading, it's in order to have a higher NDVI value, you have to have like the higher sum of the PCAs, like with that.
Well, they're separate things, but there could be a case where one of your principal components is very closely tied to like the same things that NDVI is picking up on, right? Yeah, because NDVI would increase with plant water content and chlorophyll content, photosynthetic activity. So those are also things that a principal component analysis over vegetated pixels could pick up on too. Does that make sense? Yeah, but because there's so much spectral information that's being condensed in the PCA, it could be picking up on any number of things. So it really requires like more scrutiny if you want to like really dissect each component and figure out exactly what it's picking up on. Here, I'm just more concerned about just explaining like the concept of variance and how to carry that forward and get useful information out of it. Yeah. Yeah, so that's a good question. So I guess there's two ways you can go about that. If I prefer to do everything on the mosaic, like stitch all the data together first and then do the transformation. Downside to that is the intense memory issues that can come out of that, but it's still doable. Other way you can do it is fit the principal component transformation to one flight line and apply that same transformation to the others instead of doing them all, fitting and doing them all separately. Because if you do that, you're going to be picking up on different things because the composition of each flight line is different.
Oh, so if so for example, if we
like put all the data together and figure out a PCA design that apply to all the data, we will have to manually write that PCA component for each of the data sets.
Yeah, so let's see, if we look at this line here, this fits the PCA to the image values and then transforms them separately. You could do that in one line if you want, but you could also just fit it to one image here. And then if you want to bring in other flight lines of data, you can do the transformation line on those in addition, so that it's applying the same transformation that you trained on one to all the others. That way your data would be consistent.
So,
but if you fit it separately to each one, then your results are going to look totally different.
So this Python package allow us to save the current PCA setting and apply to a different data set.
Yes, you could if you wanted.
Okay, thank you.
Mm-hmm. Another question? Okay.
Do you ever sort of, like, we made those land water masks using the NDVI earlier. Do you ever have the need, or is it like a good practice to do a land water like separation and then separate PCAs on either, or you just let the PCA technique?
Like, it depends on what your application is, but you could definitely do it that way, where you just sort of nix out the water pixels. Like if you want to just hone into only vegetated areas to, I don't know, explain more of the variance specifically within vegetated pixels only, then you might be able to pick up on more details that you couldn't by reading in all the other sort of cover types.
Yeah, I guess this is, I don't know much about PCA, obviously, by these questions, but would you be able to also just handle that by having more sort of, like, instead of 10 classes, just like adding the sort of layers in the PCA to get the same information? Or
you mean like adding the number of components?
Yeah.
It depends on the inherent dimensionality of your data. So basically, like, where you're not actually explaining any more of the variance. It could be above 10, but it would just require like more testing. I think just for a single flight line like this, overall like 10 is plenty, but it sort of depends on the composition of your image and what you want to try to explain. It could be higher.
I think most end user might not be able to process the PCA altogether, as you suggested, as the best solution to do this analysis. So will this PCA product be available along with the original data set?
So there are other libraries that do similar operations that might work better with imagery. One I use is called a Spectral Python that I find useful. It also has a PCA tool, and that's built more to actually like handle image array data. But if you're handling like a really big data set, I would also recommend Ndvi if you have access to it, because Ndvi handles these big images really well. I haven't run into memory issues, just time issues with Ndvi. Oh, it's Envi. So does everyone have the PCA file downloaded and opening Qgis now? So we're all good there?
Yeah, just a follow-up question on NDVI.
Mm-hmm.
So we're using it for the land and the water.
Yep.
So do you think if the water index, which is based on the near infrared and the short-wave infrared, could be work better for this environment?
It can, yes. NDWI, you mean, as
yes.
opposed to NDVI. So yeah, so for those who don't know, there's another commonly used index called Normalized Difference Water Index, which is based on, I think there's two different ways to go about it. It's shortwave infrared and near infrared, or shortwave infrared and green, the green reflectance.
Yeah.
I've also used that one to divide land, water, and make masks like that, and it also works very well.
Okay, so again, if we use that parameter, we're going to use do the histogram first and then find the value and then
Yeah.
define, okay.
I would use the same approach. Yes, I just didn't want to overcomplicate and introduce too much information. And also NDVI is just the most commonly used by far index for a variety of applications. So, but yes, other ones like NDWI can also be useful for that kind of use case. All right, so we have the PCA file done here. So I'll switch back to PowerPoint and we'll move on to the next module, which is just a brief introduction to classifications. So the objectives here are to perform an unsupervised k-means classification on the PCA data and generate a preliminary land cover classification image. So, and this is another example of a classification map we published with a similar approach over the Wax Lake Delta. And we'll just be sort of replicating a very, very coarse version of that with the data we've generated here so far. So unsupervised classification works by generating a given number of clusters from the input bands that you give the algorithm. So there's a few different ways you could arrive on like what bands to feed into it. It doesn't work very well to just give it 300. It kind of gets a lot of information washed out if you do that. And it's also very computationally expensive and inefficient. So you can do some sort of band selection analysis, where you look at spectral separability based on what your the targets you want that you have in mind. You can bring in additional data sources like a digital elevation model to supplement just a standard selection of bands like your typical Landsat bands, again, from this image. Or you can, again, do principal components analysis on the imaging spectrometer data to reduce the dimensionality of the data you're working with. So unsupervised classification gives you the given number of clusters that you feed into the algorithm, and then you use those output clusters and condense them into classes that you assign by referencing against some sort of imagery. So it's basically your own visual interpretation. So the most common algorithms for this are isodata and k-means clustering. And again, I bring this figure back up because it's most commonly done with platforms like Landsat, where you just feed in all the bands like blue, green, red, near infrared, short wave infrared, because those are generally non-collinear. But if you're working with hyperspectral data, those are collinear, so you do need to do some sort of band selection process. But those channels are all picking up on different sorts of information about the cover types that you might have in mind. So you can see, if we're just looking at near infrared, water absorbs almost everything. Vegetation reflects higher than soil and water. So that's the sort of information that these clustering algorithms can pick up on to like discern and differentiate those different class types that you'll then assign from the output. And just briefly, the other type of classification is supervised classification. And to perform those, you create a training data set or a spectral library that you input into the algorithm. So you typically select training areas corresponding to like remote sensing pixel data to generate spectral signatures. So if you have some known quantity that you can visually interpret, or you know from some sort of outside data source, you can plop a polygon or a point down in your GIS layer and extract all the statistics from within that layer to compile your training data set. Or the other option is if you have some like in situ data. For example, if you took a field spectrometer out and did a survey of all different vegetation types, you can compile a spectral library from that data and then use that to classify your like airborne or spaceborne spectrometer data. So some examples of this is common ones are maximum likelihood, minimum distance, or one that's very useful with AVIRIS-NG data is Spectral Angle Mapper. Basically, those work by just having some distance metric, such as a spectral angle or Euclidean distance. And so you calculate those for each pixel with reference to all your spectral signatures and assign classes that way. Machine learning approaches are also really popular for supervised classification. Those include random forests and support vector machines, amongst others. So for this module, we're gonna use the PCA output and just do a really coarse k-means clustering algorithm to make a classification of that flight line. So go back to the Google Colab and go down to part 4. So I have this commented out, but if you just delete that comment notation here, this will load the PCA results in. Mine, it's already read in from the cells above. But change this one also to just reflect where it's actually saved. So let's make sure that's in the right place.
Hey, Daniel.
Yep.
We got a question. It says, does the PCA essentially do the band selection, reducing the dimensionality of the data set? Or would you do a PCA and also a band selection?
The former. Yeah. Basically do PCA instead of a band selection because it's reducing the dimensionality and then you don't need to do anything.
Hey, Daniel, can you maybe explain that in words that those of us who don't know much about PCA might understand, just briefly?
Yeah, so band selection would be, so we have like a stack of 300 to 425 bands, and we have some targets in mind. We're gonna pick a handful of bands that are not correlated with each other that we could feed into the classification then and give us accurate results. Or we could do PCA, where we reduce the dimensionality. So we basically take the 300 bands and boil it down to the 5 or 6 bands that explain all of the variance in that original stack of 300 bands.
Satellite data, it only have few bands, the multi-spectral. Is there still any point, the value to do the PCA on satellite data?
Is there any point to doing PCA on multispectral satellite data?
Yes.
Not so much really, because the bands there generally aren't collinear with each other. I guess there are some other satellites, I'm trying to think like, that have a larger band suite where some bands might be collinear with each other, but you generally don't need to. Like for example, Landsat, I wouldn't worry about it at all.
Okay, so
what they have.
Thank you. So for hyperspectral, it's really helpful for the water. So could you talk a little bit more about the hyperspectral value for the vegetation or land application?
Yeah, so the hyperspectral value for vegetation or land applications, there's a lot more information inherent in the full spectral reflectance in each pixel than we would just capture with 5, 6, 7 multispectral bands. So we can pick up on especially like narrow features that are associated with like biochemical properties within the canopy. So like nitrogen content within the canopy can have like cause a small absorption feature in the near infrared or shortwave infrared that you can then detect with hyperspectral data. We can more accurately discern things like canopy water content than we could with just like one broad near infrared channel, for example. So there's a lot of properties that we can detect and discern and then use to produce more analysis and more accurate classifications with hyperspectral data, leveraging all of that additional data compared to just broadband multispectral images. Okay, so if we run this first cell, this first one, this imports the k-means function from the sklearn library. We generate the variable x here, which takes the PCA results where the mask equals 1 again, and puts it into the right dimensions to feed into the algorithm here. And then first thing we do is we're just going to make a simple 2-cluster k-means output. So this one, k-means equals k-means, number of clusters is 2, and then we'll just fit and predict on the same line. So this will basically essentially give us the same output or very similar as the NDVI-based water mask, but this is just to give a sort of quick and dirty first example of the of how the classification works. So then we could plot the result, and you can see 0 is water, 1 is land. It looks very similar to that NDVI-based mask, right? Does that work on everyone's computer? I just want to make sure we're... Okay, yeah, so I have this one line that's commented out. So you'll need to make sure this is pointing to where the that PCA file is saved first. Yeah, so you can replace this section here to point it to this file.
On the Google Drive.
On the Google Drive. And then
Not
it'll
on your
read
new
that pca.tiff in. So I had this, I had this output directory set to that folder in mind. So that's why that's stayed like that.
So for you all, it should be input _ dir. Maybe you can type it out, Daniel. Maybe you can type it out.
Yeah.
I'm just saying it.
So it's the
should be the same as the input, but then / output.
Yeah. Tutorials, AVIRIS-NG. Let me make sure at the top here. Yeah, here we go. So this is where I had it set before.
That's for the offline people, though, right?
Yeah. So let me just copy this over.
Oh, no, because you want it for Google Collab. Shouldn't it be this one?
Oh, yeah, you're right.
So you can just set it equal to input dir. And then add in the / output. So yeah, I think the PCA file is saved on the Google Drive in a folder called output that is within this tutorial. So if you change, like you just did, except I think you need to add, you can put it maybe here, the / output.
Here, we'll just make it simple here. AVIRIS-NG /data /output. And then file there.
Okay, can everyone see that?
You can manage your paths however you prefer. Some people join them with a +, something like that. You could just type it all out in one go.
Do you not have access to the folder? It says empty. Oh. And it worked? Okay, good.
Yeah, sure.
Raw data I saw, it is HDR format. Do you know, is there any tool to convert it to TIFF or something we
The
can
header
read in?
file?
Yeah.
So the header file is metadata that's associated with the binary file.
So what's the format of the raw data? I don't see the.
So it actually is rather important because it contains the like projection information for the image.
Yeah, the raster is TIFF format.
Yeah, but I have been outputting all of our results as TIFFs just so that
Okay.
we don't have to like handle both types of files, and it works best generally with GIS.
So I don't know if Google Collab, like, if you are gone for too long and it disconnects, you may need to go back and rerun the first two cells where you import your packages. I think that's why it asks you if you're still there.
Question? Yeah, so so the input data that we're giving it is the 6-band PCA data, right? Oh, so he just asked me to explain k-means clustering a bit. So that's the input along with the number of clusters we want it to output. So basically, it just projects the multidimensional input data, so the six-band data, onto some two-dimensional space and finds the clusters, the cluster centers for like two clusters that you want it to output. And then it sort of reprojects the data and assigns like which of those cluster centers is it closest to. And then it groups it that way. So if we assign it more classes, so in the next cell, we'll tell it to give us 10 outputs. It'll find the 10 most distinct cluster centers. And for each pixel, then it'll say like which cluster is it closest to. And then we'll take those 10 outputs and we'll like try to assign the actual like class meaning to them to make a classification map. Because it'd be more work for the purpose of this class, more manual generation input of data. This is a better starting point. I also find unsupervised classification in a lot of cases to be more useful for this kind of data structure. It can also be, I also work with some supervised classification types based on like spectral libraries. So that could be very useful and works well with AVIRIS-NG data, but it's just a little much for right now. Yeah. All right, so are people getting there with the that first just 2 cluster output? Cool, seems so. All right, so we'll just go on to the next one. It might take a minute to run, but basically, again, it just does the exact same thing, except we feed it, make it give us 10 clusters. And then it'll give us the output. And given that it takes a few seconds in this computer, I just hope it doesn't crash anyone else's on Collab, but it should be fine. Yeah, so
Can you repeat that question?
you're asking about this line here where class map where mask equals 1 equals k-means. It's because the k-means output is a one-dimensional output. There's like 1 entry for every pixel that we ran it on, but we're just feeding those values back into the mask to like make a map out of them. So I'm just like, we took it, we made it like one-dimensional to like feed into the classification algorithm, and we're just like putting it back into 2 dimensions to like turn it back into a map, if that makes sense. That's why we use the mask function a lot. It's just for indexing purposes, handling those values. No, well, so the actual output of fit predict is the index value of which cluster the pixel belongs to. Exactly. Mm-hmm. So that finished running here on mine at least, and you can see the output here. It's not very friendly color scale for the raw output, but you can see that it assigned different class or cluster outputs here for the map. So then our next job is to like decide what those mean and assign them actual classes.
Got it. So my question is just, I want to repeat my question, is that the color map in this figure is from 1 to 10 representing their index in their cluster.
Yes, that's right. All right, so when that's done running, some of yours might not be yet, but I'll just run the next block, which again just saves the output, which you'll then want to download. And then what we usually do is bring it into GIS and just reference it against like a true color image or a false color image if you have one that also sort of helps you discern what land cover types are, and then figure out how to assign each of these different class outputs. So, I mean, first things that pop out obviously are 0, and I think, what is that, 7? Yeah, 0 and 7 are water classes, right? And then this blue color looks like it's green vegetation. There's some like non-photosynthetic vegetation looking patches here as well that line up well with this one. So basically when you take this cluster output, you just sort of go one by one through each class, look at the features on the ground, figure out what they mean. They don't always line up like as cleanly as you want them to, like invariably. So you have to embrace a little noise. There's going to be some areas of confusion, but you generally just try to figure out what sort of class type each of these cluster outputs best belongs to. So the way I have it, I said before, it's very rough classification. So I just have four classes here that I've assigned: water, tidal mud flattened soil, senescent or non-photosynthetic vegetation, and green vegetation. So I would just take a couple minutes in QGIS and scan around the image. You can play with the colors if you want to try to help you group these clusters. And then I go back to the code and actually like reclassify those numbers into that output. Yep.
Well, to show how to combine those two classes in QGIS?
There's probably a better way in QGIS. Maybe use like a raster calculator function or something like that. Easiest way, I think, is to just change the colors manually. So 0, we know is water.
Yeah,
so we could just,
got it.
we could just change the ones we know one by one.
Yeah,
Colors
I just
like
trying to make sure the QGIS is similar as ArcGIS or GIS Pro.
Yeah.
So do you know what's the major difference between those software?
Between QGIS and ArcGIS?
Yeah.
I don't know that I don't think there's that much difference. QGIS kind of gets better with time, so I think there's a lot of overlap in functionality.
Okay.
So you guys should have probably, since we have the same parameters and the same input data, you should probably have the same output clusters that I do. So you could just use this scheme that I wrote up, or you could change the numbers if you want, if you think something is misplaced or you would like to add a different class. It's totally up to you. So my output, and again, is a rough classification, but it lines up pretty well with what's on the ground for just these 4 very broad classes. Looks like this.
So if you're not able to get it to output from your notebook, either because the notebook has reset and all of the things we fixed don't work anymore, you can just download this classification from the Google Drive, correct?
Yep. Yeah. I uploaded this one up there too. So you can take a look.
Yes. So as soon as your collab is tired and resets, those things like changing the output directory are gone, and fixing the other file locations are gone. So you would have to redo those if you wanted to work again. Just a heads up.
Yeah. So, I mean, you could just double click on them from here if you want. It'll take you into the color scheme. Oh, I'm sorry. Make sure it's set to paletted unique values here.
The question is how to get the colors for one band.
Yes. Yeah, so click paletted unique values, and if they don't pop up, automatically hit the classify button down here. And then it should identify all the unique values present in your image that you can then assign. So just one final note for classification results broadly. You can always adjust the k-means parameters, such as the number of output classes, if you want to try to classify more. You can set it to like 30 output classes to try to get more output classes that it'll pick up on. And again, you can also change your input bands, including the which PCA components you've selected. You could add the NDVI data or other bands or spectral indices in there as well if you think it'll benefit it, or other geospatial data like elevation or something like that that you think might have an impact on what classes you're trying to classify. So from my own experience, for example, I found elevation helped, adding elevation onto principal component analysis bands helped sort of identify different vegetation types that were sort of stratified by elevation. So we could really separate species that belong in different elevation zones by introducing that other variable.
Maybe now would be a good time to survey how people are feeling. Are we feeling like we're falling behind? Are we keeping up? Are we lost entirely? Are we way ahead?
There's just one more.
Does anyone need a break? Guess not. Okay.
All right, so people think they have the handle on the classification and have the output more or less. All right, so we'll move on to the last one, which is on partial least squares regression and water quality. So for this module, we use in situ water sample and reflectance data to generate a regression model for suspended sediment concentrations or total suspended solids and create a map of TSS with the flight line here. So this is an example of this approach we applied to the pre-Delta-X campaign data from 2015 and 2016.
There's a question online. Have you been able to calibrate the k-means result or validate the result?
The one that we just generated or the previous example that I had showed?
I'm thinking the previous example.
Okay, so if we're talking about just the flight line results that we did the rough one before, I mean, I didn't go over how to validate or anything in this one. If it's rough classes, you could probably just generate like a random sample of points across your image and identify whether they're true or not just from imagery on your computer. If you get down into coarser detail, like where you're trying to like identify vegetation species or other types that aren't really interpretable from just an image, you really have to go on site. So for example, to validate this one, we identified some transects. I pulled Alex and Andre through the mud with me to get those validation data sets. And yes, we just validated it on site with 100 some points to get an accurate representation of all the classes.
This is Mark online. I'm not sure if you can hear me, guys, but
we can hear you.
Yeah. So yeah, I want to make sure people understand. Maybe I should have mentioned that in the introduction, but everything you're seeing today is fresh off the grill. You know, we just we spent the last few months preparing these data sets, but they have not been analyzed yet. So what you're seeing today is really preliminary analysis with no conclusions or validation. So this is really fresh data. So at this point, you're up to speed with us, and yeah, we hope to have validation and assimilation into models much later. But yeah, for now, this is all new.
Yeah, so for all our L3 products like vegetation classifications, biomass maps, and sediment concentration and whatnot, those will be validated with a combination of additional study, but also field data that we've already collected. But those products are generally all still in progress. So what we're presenting here is just sort of test cases with the remote sensing data that we have right now.